Okay, so welcome to this video. In this video, what we're going to talk about is angiogenesis and angiotherapy. Okay, so let me give you an overview of the structure then for this video. Okay, so we're going to start off with a basic discussion of what angiogenesis is. Okay, so we'll look at a broad overview of the process of angiogenesis. Specifically, we'll look at angiogenesis in response to a hypoxic area of tissue, okay, rather than other examples which we could use, which are angiogenesis in the endometrium within the menstrual cycle, and also angiogenesis in areas where granulation tissue is forming in wound healing. Okay, we're going to specifically look at the pathway for angiogenesis in the context of a hypoxic area of tissue. Okay, right. Then after we've done the broad overview, what we'll do is look at the process in much more detail. So we'll look, for instance, at the mechanism by which cells which are exposed to hypoxia actually upregulate their expression of pro-angiogenic factors, such as vascular endothelial growth factor and fibroblast growth factors as well. Okay, so we'll look at that mechanism in detail. Then what we'll do is discuss what the vascular endothelial growth factor and fibroblast growth factors do to the endothelial cells of capillaries to cause them to produce these sprouts. Okay, then we'll finish off the process of angiogenesis. Okay, then what we'll move on to is discussion of angiotherapy. Now, angiotherapy is just a word that means using drugs that target angiogenesis, i.e. alter the process of angiogenesis, either inhibit it or potentially stimulate it, as clinical therapy, basically. So that's all angiotherapy means. Treatment that involves targeting angiogenesis. Okay, and we'll specifically look at angiotherapy for cancer. There is a cardinal example of a drug that is used for inhibiting angiogenesis to cancer, which is bevacizumab, and we'll discuss that drug later on. Okay, then we'll also look at angiogenesis' role in diabetic retinopathy, because it plays an absolutely key role there. And again, diabetic retinopathy can be treated with angiotherapy. Again, this drug, bevacizumab, the trade name of which is Avastin. Okay, and we'll have a look at what underlies diabetic retinopathy and how angiogenesis is involved in that, and therefore how bevacizumab is going to be effective in treating diabetic retinopathy. Okay, then finally what we'll do is talk about something potentially for the future, which is using angiogenesis to treat coronary heart disease, and in that case we want to upregulate the angiogenesis. In the two previous cases, angiotherapy for cancer and angiotherapy for diabetic retinopathy, we want to inhibit the angiogenesis. For coronary heart disease, we'll want to increase angiogenesis. So we'll look at potentially that as a potential treatment for the future, basically. Okay, right, so let's start off with a few definitions. Okay, so angiogenesis is the creation of new blood vessels in the adult human. Okay, so the growth of new blood vessels in an adult human. Okay, there is another word that means the exact same thing, which you'll occasionally hear people using, which is neovascularization. Okay, so neovascularization again just means the production of new blood vessels in the adult human. Okay, now both of these words mean the same thing, but they are absolutely not the same as another concept, which is the concept of vasculogenesis. Okay, so vasculogenesis, the word, if you just look at the word, you'd think maybe that means the same thing as angiogenesis and neovascularization, but it doesn't. Vasculogenesis refers to the actual creation of blood vessels in an embryo. Okay, so from precursor cells actually producing blood vessels from scratch in the embryo. We are not going to discuss vasculogenesis at all in this video. We are going to concentrate on angiogenesis, which is also called neovascularization, which is the creation of new blood vessels from existing ones is the key thing here. We're not just creating them from scratch. We are just basically making little extensions of existing ones. Okay, so that's the process we're going to look at, angiogenesis, also called neovascularization. So where is angiogenesis important in the adult human? Well, I'll start off with physiological examples of where it's important, and then we'll discuss some pathological examples, and I've already hinted to those in the structure discussion that we've just had, but I'll outline them again when we come to pathological involvement. Okay, so what physiological scenarios is angiogenesis important in? Well, the key ones here are it's involved in producing new blood vessels to reach areas that are hypoxic. Okay, so if you have areas of tissue which are hypoxic, which I'll just refer to as hypoxic tissue, you can get angiogenesis occurring to produce new blood vessels that will go to that hypoxic area and will deliver a blood supply to that area, and therefore it will no longer be hypoxic, is obviously the key there. Okay. In addition, angiogenesis is extremely important in wound healing, the normal healing process for a wound. Okay, we won't actually look at the exact pathway of wound healing in this video, but the pathway that we're going to look at, which is for hypoxic tissues, a lot of the same things happen in the wound healing pathway as well for angiogenesis, just with a few minor alterations. Okay, and then finally, another key involvement of angiogenesis in the adult human is in women who are going through the menstrual cycle. Okay, so when the endometrium grows in preparation for the implantation of the embryo, the new tissue that you have in the endometrium needs a blood supply, and this means that we have to grow new blood vessels in the endometrium, and this is done by a process of angiogenesis. Again, we won't be looking at the angiogenic pathways specific to the menstrual cycle, but again, there are a lot of parallels between what we will discuss and what occurs there as well. Okay, right. So those are physiological examples of where angiogenesis is important. Okay, let's now mention the pathological examples of where angiogenesis is key to the pathological event. Okay, so pathological scenarios. So we've mentioned cancer and diabetic retinopathy, so those are the two key ones. Basically, tumours, as they grow, they're going to need a new blood supply, basically. In fact, tumours can't get beyond 1 or 2 millimeters in diameter without angiogenesis occurring. Okay, so 1 or 2 millimeters is roughly the size a tumour can get to without any more angiogenesis, meaning that that's the distance over which nutrients can diffuse and waste products can diffuse without actually getting a new blood supply infiltrating into the tumour. Okay, so 1 or 2 millimeters isn't that bad, so if we could maintain it at that by inhibiting angiogenesis, then obviously that's the great hope for how we could treat cancer. Okay, and we'll come on to that much later. So those two both involve angiogenesis big style, basically, and as I say, right at the end we'll look at angiotherapy for both cancer and diabetic retinopathy, because there is a drug that is now used clinically which it does exactly this. It inhibits angiogenesis, and it's used in both of these conditions. Okay, right, so that's an overview of where angiogenesis is important in medicine. Okay, let's now talk about the actual process of angiogenesis itself, the actual pathway of angiogenesis. Okay, and as I say, we are going to look at the pathway for a hypoxic tissue. Okay, so we're going to have a portion of tissue that's hypoxic. It's going to cry out for help, basically, and this is going to trigger blood vessels to sprout new blood vessels off the side of themselves, which will then converge on this hypoxic centre, and then the blood vessels that are converging on this hypoxic centre will then fuse together to make a full blood vessel. Okay, and I'll do a broad overview of that in a moment. Right, so before we start anything, I just want to make sure that everyone is familiar with the microcirculation, specifically what the structure of a capillary is, because that's going to be extremely important throughout this entire discussion. Angiogenesis is everything to do with the capillaries. Okay, it's the capillaries which are going to take part in angiogenesis, not the massive great arteries. It's the capillaries that can have little sprouts coming off them. You can't get massive great sprouts coming off your aorta. Okay. Okay, so let's just discuss how the microcirculation works. So just a basic recap of the microcirculation physiology. Okay, so you have a very small little blood vessel here now, which is called an arteriole. Okay, and this is bringing arterial blood into the area. Okay, now this blood vessel will have smooth muscle cells lining it, and the smooth muscle cells will control its diameter, okay, and it can either vasoconstrict and reduce the amount of blood flowing into this area, or it can vasodilatate and increase the amount of blood flowing into this area. So the arteriole is the key blood vessel that controls how much blood flows into a little local area, basically. So we'll colour in the arteriole in red. Now what's going to happen is the arteriole is going to split up and branch into loads of much smaller blood vessels, which are the capillaries. Okay, so I'll show these now. So here they are. This is the capillary bed here, multiple capillaries all running in parallel like so. And then they'll reconverge together to form the venule here, okay, which is again a much bigger blood vessel than the capillaries, but still a very tiny blood vessel. You know, these aren't the massive great arteries and veins that you learn about in anatomy. These are tiny little blood vessels. Okay, so here's the venule in blue, and the blood vessels we're going to be specifically interested in are the ones in the middle here, the tiny little ones. Okay, the capillaries, which I'll colour in now in green. Okay, so these are the capillaries. Okay, so what we now want to do is have a look at the structure of a capillary. So what I want to do is look at the cross-section of a capillary. So imagine that we cut through a capillary, and now we're going to look at the cross-section. Okay, so basically capillaries have two main cells forming their wall. Okay, they have endothelial cells which are sitting on the basement membrane, and then around the basement membrane they have pericytes, but it's not a confluent layer of pericytes. Okay, the pericytes are dotted around on the edge of the capillaries. So at certain portions of the capillary wall, all you'll have is the endothelial cell with the basement membrane underneath it. Okay, so let me draw this here. So here is an endothelial cell here. And capillaries really are tiny little blood vessels. The little tube going down the middle, the lumen of the capillary, is pretty much only wide enough for a single blood cell, red blood cell, to move through, or a white blood cell. Okay, so here are a few endothelial cells here lining the wall of the capillary. Okay, and these are their nuclei, and I'm not sure why this one's nucleus ended up so much smaller than that one's. Okay, right, so those are the endothelial cells, and I will abbreviate endothelial cell continuously down in this video just to EC. Okay, but I suppose I should write it out in full at least once. So E stands for endothelial, okay, and the C stands for cell. Okay, right, so then underneath the endothelial cells, and what the endothelial cells are attached to, is a basement membrane, which I'll colour in turquoise here. Okay, so the basement membrane is a layer of proteins. It's a meshwork of proteins, mainly collagen, but also other proteins besides collagen. Okay, notably laminins as well. Okay, and the endothelial cells are going to be attached onto the basement membrane, and this is what is keeping them in their position and stopping them effectively dropping into the lumen. Okay, so they are attached to this rigid protein meshwork that lines the entire blood vessel. Okay, right, so this is the basement membrane here, and again I will be abbreviating basement membrane down to BM throughout most of this video, so I'll just write it out in full once. So BM for basement membrane. Okay, right. Now, outside of the basement membrane, you have another cell type that's sort of dotted around the place. So as I said previously, it's not a confluent layer of cells that you're going to have outside the basement membrane. What instead you're going to have is them sort of dotted around different places. So here's one, okay, and then you might have another one over here maybe. Okay, and these cells are known as pericytes. Okay, so I'll label it up. So this is called a pericyte. Okay, peri means around, cyte means cell. So it's a cell that's around the capillary, basically. Okay, and I think I'll colour in the pericytes in blue. Okay, now these pericytes have the ability to contract, okay? And when they contract, what this can do is it can, to an extent, constrict the capillary lumen. Okay, so when they contract, it can end up squashing the capillary lumen down a bit and constricting it. Okay, so to an extent, capillaries can vasoconstrict and vasodilatate by virtue of their pericytes being able to contract and relax. Okay, right, so that's the essential stuff that we need to know about the structure of capillaries to progress further with this discussion. Okay, right, so what I'm going to start off with is a very broad discussion of what's going to happen in hypoxia-induced angiogenesis. Okay, so I'm going to go over the whole process in much more detail in a moment, but for now I just want to give the basic overview of what's going to happen so that we know where we're heading towards, what is actually going to happen, and then we can look at the in-detailed mechanisms of actually how it happens. Okay, so this is the broad overview, so I might just put that as my title here. So this is a broad overview, and we'll go into the actual details of this in much more detail in a moment. Okay, right, so let's say this is one capillary here. Okay, so this is a tube, this is a capillary here, and then we've got another capillary here. Okay, and then let's say in the middle here we have an area of cells that is hypoxic. Okay, so this is our hypoxic center or our hypoxic area. Okay, here. Right, so basically what's going to happen is if these cells become hypoxic in this hypoxic area, they are going to start sending out screams for help. Okay, they're going to start sending out all sorts of molecules which are basically screams for help. Okay, and these molecules are going to go over and act on this capillary over here, and also this capillary over here. Okay, and the capillaries are going to respond through angiogenesis, basically. Okay, because after all, the tissue is hypoxic. What does it need? Oxygen. How can we supply oxygen to it? Well, by giving it a better blood supply. Okay, so we're now going to grow a new blood vessel towards this hypoxic area. So what's going to then happen is this is a sort of arrow showing from number 1 to number 2. So this is picture number 1, and now we're going to go to picture number 2. So this is going to progress towards this situation. I've just ruined this. I was meant to draw a sprout coming out here. Never mind, I'll just have to do it unprofessionally like so. So ignore that line there. Okay, so that's one sprout coming off that capillary, and now I'll do this one better. Okay, like so. Here's this other capillary with another sprout coming off it, and now you're getting your hypoxic area here, which will still be sending out those molecules. Okay, and you can see that these two capillary sprouts, as they're called, okay, so these little projections of the capillaries outwards, these are known as the capillary sprouts. Okay, the capillary sprouts are going towards the hypoxic area. Okay, and then the idea is that when these two capillary sprouts eventually get there, they're going to fuse to make one confluent vessel. Okay, so let me show this. Like so. So here's the first capillary, and now it's going to be in a sort of H shape here. Okay, and we've now got our new blood vessel, and that will be going right through that area that was previously hypoxic. So there is no way that that area can be hypoxic anymore. It's got a beautiful blood supply now. Okay, and therefore we have sorted out the problem. Okay, so this is the broad overview of what is going to happen in this ischemia-induced angiogenesis. Okay, we're going to get these capillary sprouts coming from neighbouring capillaries. They're all going to be converging to this hypoxic area, and of course you could have more neighbouring capillaries that are giving capillary sprouts than just two. We're going to leave it at just two because it's simple to understand with just two, but you could have loads of them all converging together and forming a chaotic meshwork, basically. Okay, and when they converge to this hypoxic area here, they're going to fuse together, and then we've got this nice blood vessel now connecting our two capillaries, and blood can move down here into here, or potentially down here into here, depending on which capillary has the higher pressure. Okay, right, the point is that blood will flow through now, and you're now going to have a very decent blood supply to that area there. Okay, so it will no longer be hypoxic. Okay, so that's a broad overview of the process that we are about to study. Okay, so we're going to start off then with the discussion of what are these screams basically, and how are they produced. Okay, so when an area becomes hypoxic, what does it actually release to trigger the angiogenesis process? What does it release to trigger these capillaries to produce these capillary sprouts? Okay, and the main answer is vascular endothelial growth factors. Okay, so we're going to go to vascular endothelial growth factors first. Okay, so for short, vascular endothelial growth factor is abbreviated down to VEGF. So I'll write out its full name once. Okay, so the V is for vascular, the E is for endothelial, okay, and then the GF, as always, is for growth factor. Okay, now, the vascular endothelial growth factors are actually an entire family. Okay, so let's discuss the family first, and then I'll say, when people usually say vascular endothelial growth factor, there is a specific member of the family that they usually mean. Okay, so we need to discuss all of this nomenclature so that we're all clear with what I mean when I say vascular endothelial growth factor. So really, there is a family of proteins which are called the vascular endothelial growth factor family. Okay, and in this family, there are 5 different proteins. Okay, so what are these 5 different proteins? Well, there is vascular endothelial growth factor A. Okay, there is also vascular endothelial growth factor B, and I won't rewrite out the VEGF portion. Okay, there is also vascular endothelial growth factor C, and there is vascular endothelial growth factor D. Now, unfortunately, the 5th and final member is not called vascular endothelial growth factor E. Instead, it's called placental growth factor. Okay, and this is written PLGF, like so. So this stands for placental, that's the PL. Okay, placental, and then the GF is just for growth factor. Okay, so that's also in this family of vascular endothelial growth factor proteins. Okay, right, so what do all of these proteins share in common? Well, basically, they all act on their receptors as dimers. Okay, so all 5 of these proteins form homodimers, like so. So basically, if you have, let's say, a vascular endothelial growth factor A protein, what happens is it dimerizes with another identical vascular endothelial growth factor A protein, and then the homodimer, like so, is the functional unit. This is what actually binds to the receptors for vascular endothelial growth factor and will actually activate downstream pathways within cells, as we'll discuss in a moment when we come on to the receptors for these. Okay, so all of these proteins form homodimers basically, and they act on receptors as homodimers, okay, rather than as single proteins. And in fact, what happens is the homodimer goes up to the receptor, and one of the members grabs one receptor and the other member grabs another receptor, and that causes the receptors to dimerize as well, but the ligand itself is pre-dimerized before it actually binds to the receptor. Okay, contrast that to, for instance, the epidermal growth factor, which isn't pre-dimerized when it binds to the receptor. Okay, right, so now let me tell you about the fact that when people say vascular endothelial growth factor, okay, so when they just say VEGF, what do they mean? Okay, well, they don't mean all 5 of these. They mean specifically vascular endothelial growth factor A. Okay, so if someone just says VEGF, without clarification as to which one they mean vascular endothelial growth factor A. Vascular endothelial growth factor A is the most important one in angiogenesis. Okay, so it's the main one that these hypoxic areas will be releasing, and it's the main one that forms their screens, basically. Okay, right. Vascular endothelial growth factor B and placental growth factor, they are very involved in embryology again. Okay, so they're involved in vascular development in the embryo, and we're not going to really mention those at all. Okay, so they're involved in embryology. Finally, the other 2, vascular endothelial growth factor C and D, these are also important in angiogenesis, so they also participate in this. They will also be part of the screens that the hypoxic area releases. However, their role is nowhere near as important as vascular endothelial growth factor A. So we're just going to sort of shove them to one side and we'll just talk about vascular endothelial growth factor A. Remember that they're also there and they're also participating, but this one is the most important one basically. This is the main one that these hypoxic area cells are going to be releasing to signal to the endothelial cells of the capillaries that they need to produce these capillary sprouts. Okay, right. So now what I want to turn my attention to is how do the cells in this hypoxic area actually start producing vascular endothelial growth factor A, which from now on we'll probably just refer to as vascular endothelial growth factor. Okay, and it's that question that we'll turn our attention to in the next video.